Hello, good evening. It's myself is Mr. Rasul. I have 18 years of experience in the NEET. I can teach to the biology. Okay. Today it's my concept is a C3 cycle in the photosynthesis. Why I am choosing the C3 cycle means nowadays the population day by day increase. How we will get the food materials from the plants? The major source, the ultimate source from photosynthesis only. That's why the C3 plants or C3 cycle final agenda is to preparing of carbohydrates. The more than 800 people, 800 crores of the people are there in the worldwide. How we will get the food? Mean especially due to photosynthesis and what type of cycles are there? C3 and C4 cycle. But today it's my concept is a C3 cycle. And before I want to explain this C3 cycle, first of all, we will discuss about light and dark reactions. What is the light reactions? When the plants, the receiving of the sunlight, especially where it will be focusing on the sunlight, mean the green parts of the plants. In the green plants, why it is a green in plants mean the chlorophyll and chloroplast structures are there in this region. Especially, you take the chloroplast structure. The chloroplast is a double membrane layer. The central place, the inside of the chloroplast, granulothylakoids are there. These granulothylakoids is connected to these granulothylakoids connect to the Fred's lamella or stroma lamella we call, and especially sunlight where it will be focused in this granulothylakoid. So one sunlight it will be forming. Like which purpose I am saying it is the light reactions mean the final products of light reactions ATP, NADPH and oxygen. These three are main products. Oxygen is a byproduct. This product only we are getting, we will take like majority of all the living organisms dependent on oxygen only. What about remaining product? That is the ATP plus NADPH I told. This ATP NADPH is a from this granulothylakoid to where it will becomes in stroma region. The stroma region only we observe in the dark reactions. In the dark reactions we are divided into mainly two parts and two cycles are there, C3 cycle and C4 cycle. So why we divided into the C3 and C4 cycle, especially the based on the primary product. When the primary product it will be formed in the C3 cycle, that is the PJ is there, phosphoglyceric acid it will be formed, that is the 3-carbon compound, that's why we call it the C3 cycle. In C4 cycle, the first stable compound is a OAA, oxaloacetic acid, it is a 4-carbon compound. Okay, today it's my topic is a C3 cycle, especially where we observe, first of all, whom given this one? This concept was given by, his name is a Melvin Calvin. Melvin Calvin. That's why we call it the Melvin Calvin cycle also. Not only C3 cycle, Melvin Calvin cycle. Okay, sir, which plant was observed this Calvin cycle? Chlorella is a one very good algae there. In that algae, C14 isotope was used. The based on that isotope only was clearly analyzing of the C3 cycle, how the carbon dioxide to receives, how the carbohydrates to prepare inside of that stroma region, and extraordinary information was explained Mr. Calvin. Now, it was given 3 steps are there. Mainly, we are divided into C3 cycle. 3 steps are there. The first step, what we call carboxylation. Carboxylation. The second one, reduction. The third one, regeneration. And these are the main 3 steps. Okay. You'll see this is the carboxylation mean the outside. When we release the carbon dioxide, this carbon dioxide and water molecule first receiver is a cumene. RUBP, ribulose bisphosphate is present in the stroma region. This RUBP only first carbon dioxide receiver. The 5 carbon, the RUBP is made with 5 carbon compound. Once carbon dioxide binds with RUBP in the presence of rubisco. Rubisco is one of the abundant protein. It is one of the enzyme in the plant enzyme. Ribulose, bisphosphate, carboxylase, and oxygenase. Like 2 names, the multiple character showing of this enzyme. Okay. IUPAC binds with carbon dioxide. Its first stable compound is a phosphoglyceric acid. This 5 carbon compound and another one carbon is there, like 2 molecules of PGA it will be formed. That's why here 6 carbon compound it will be prepared. After that, like this PGA is a conversion into phosphoglyceric acid is conversion into triose phosphate or TP we called. This triose phosphate preparation in between, here are 2 ATP energies to utilize and formation of 2 ATP and inorganic phosphate. At the same time, 2 NADPH2 to utilize here. 2 NADPH to utilize. Like here, the finally the PGA is conversion into triose phosphate in between to utilizing. The PGA is conversion into triose phosphate. This process what we call reduction process. Like finally, here in the triose phosphate, especially here 5 carbon, 6 carbon compound is there. Here 6 it comes in, like in the 6C only 1 carbon is there. Here 1 C it comes. This cycle 6 times it will be done. 6 carbon dioxide to utilize. Then here how many carbons been? A 6 carbon compound related. Then glucose it will be formed. Already you know glucose formula C6H12O6. This is the final product. At the same time, again the process, the cycle is a regeneration purpose. One more step is there here, regeneration. The triose phosphate is there. Here 1 carbon dioxide is coming in 5C carbon. Like this, triose phosphate again it will be regenerate and its formation of ribulose bisphosphate. In this region also, to utilizing of 1 ATP energy. Overall, 1 carbon dioxide fixation purpose. Mean 1 carbon dioxide fixation purpose, how many ATPs are required? 2 ATP in the reduction area, 1 ATP in the regeneration area. That's why in the examination point of view, many times they ask the question, 1 carbon dioxide fixation purpose, how many ATPs are required? 3 ATPs are required. 1 carbon dioxide fixation purpose, how many NADPH2s are required? Mean 2 NADPH2s are required here. 2 NADPH2s are required. Here, overall, this only just I am saying here, reduction process area 2 ATPs and here 1 more ATP, total 3 ATPs are there. But here 6 carbon dioxide fixation purpose, how much we want? 6 CO2 fixation purpose, just you should calculate 6 into how many other 3 ATPs mean 6 3s are 18 ATPs are required to completion of this cycle. At the same time, how many NADPH2s are required? Mean 6 into 2, 12 NADPH2s are required to completion of C3 cycle. Okay, this is the final product of in the completion of C3 cycle. What are the final products? Mean carbohydrate or starch or glucose. Like in the different brands, just you can take the sugar cane. Like it is the best example of C4 cycle. Sugar cane we will get like sucrose. The C4 cycle, it is a quite different anatomy is there, like that is the concept, that final agenda also to preparation of carbohydrate material only. Okay. Thank you very much to giving this opportunity. Okay, bye.